Welcome to this deep dive. If you've ever looked in the mirror, maybe spotted a new gray hair or felt a weird ache in your knee and wondered what is actually happening under the hood as the years tick by, you are in exactly the right place.
Yeah, absolutely.
Because today we have a very specific mission. We are exploring a monumental, brand new study straight out of Rockefeller University. It's titled Organism-wide Cellular Dynamics and Epigenomic Remodeling in Mammalian Aging.
Quite a mouthful, but yeah.
Right. But the goal of our deep dive today is to unpack exactly what happens to our cells as we age. We want to understand why men and women experience aging fundamentally differently at a molecular level, and really see how this specific research could completely revolutionize future therapies.
It's a genuinely groundbreaking paper. It fundamentally changes the narrative we have around growing older.
Yeah.
Because I think a lot of people naturally think of aging as just general wear and tear.
Yeah, like an old car rusting over time.
Exactly. You imagine the parts just slowly wear out, the engine gets sluggish from decades of use. But what the data in this study suggests is that aging isn't just random degradation. It is a highly orchestrated, body-wide rewiring of our DNA's instruction manual.
Which is wild.
It really is. It's not passive decay. It is an active, systematic reprogramming of how our cells operate.
Okay, let's unpack this because the sheer scale of the source material we are looking at today is completely unprecedented. To even make a claim that bold about how aging works, you need an astronomical amount of data.
You do.
And the researchers didn't just look at one specific organ or, examine a few isolated cells in a petri dish. They essentially built a massive atlas. Can you walk us through the actual scope of what they did here?
Yeah, so the scale is really what makes this paper so definitive. They built a single-cell atlas by profiling nearly 11 million individual cells.
11 million.
Right, to put that in perspective, early genetic studies might have looked at a few thousand cells. 11 million gives you a high-definition panoramic view of the entire biological landscape. And they pulled these cells from 21 different mouse tissues.
So we aren't just looking at the heart or the brain, we're looking at everything. The liver, the gut, the lungs, the skin.
Everything.
Yeah.
And to really understand the trajectory of aging, they didn't just look at old mice.
Right, they had to map the journey.
Precisely. They looked at these 11 million cells across 3 distinct life stages. They started with young mice at 1 month old, then adults at 5 months old, and finally aged mice at 21 months old.
And they did something else that was crucial, right?
Yes. The most crucial decision they made, which is something biological research has historically failed to do, was ensuring they looked at both sexes in equal measure.
Ah.
And as we'll get into later, separating the male and female data turns out to be one of the most revealing aspects of the entire study.
I cannot wait to get to the sex differences because my mind was blown by those numbers. But before we get there, we need to talk about how they looked at these 11 million cells. Because they weren't just taking a roll call of what cells were hanging around.
No, they used a cutting-edge technology called EZ-Sci-ATAC.
EZ-Sci-ATAC, and they used it to look at something called chromatin accessibility. Now, for anyone listening who hasn't taken an advanced biology class recently, how should we visualize this?
It's a dense term, but it's the absolute core of the paper. Think of it this way. Every single cell in your body has the exact same genome. The DNA in your skin cell is identical to the DNA in your liver cell.
Right.
So how does a cell know to act like a liver instead of a patch of skin? It comes down to chromatin. Chromatin is the complex of DNA and proteins that packages your genome inside the cell nucleus.
Okay, let's use an analogy here to make it stick. Imagine your entire genome is a massive, multi-volume instruction manual. Because every cell has the same manual, they all have the same potential instructions. But a liver cell and a brain cell need to read entirely different chapters to do their specific jobs. So chromatin accessibility is essentially the physical state of that manual. It tells us exactly which pages are tightly glued shut and which pages are forced wide open for the cell to read at any given moment.
That is the perfect way to visualize it. By measuring chromatin accessibility, these researchers weren't just seeing what a cell was doing. They were looking at the physical control switches.
What's allowed to do.
Exactly. They were seeing what the cell was allowed to do by its own DNA packaging. And the shocker here is that as the mice aged, the physical structure of that instruction manual fundamentally warped. Pages that used to be open slammed shut, and pages that were safely locked away ripped open.
Which leads to a massive biological game of musical chairs. When the instruction manuals start changing, the populations of cells in the organs start shifting dramatically. I was looking through the data, and it says about 25% of the 536 organ-specific cell types they identified undergo massive population shifts as we age.
25%. It is a total restructuring of the body's workforce. And we can really divide this shift into the cellular losers and the cellular winners of aging.
Let's start with the losers because this part felt very personal to me. These are the highly specialized functional cells that literally start disappearing from our organs, causing us to lose our regenerative capacity. I was reading the section on muscle tissue, and it seemed like the decline starts way earlier than I expected.
Yeah, it really does.
When exactly are we losing these repair cells?
That is one of the most sobering findings in the atlas. You would intuitively think that cellular loss is something that only happens in the aged 21-month-old mice. But we are seeing crucial cells like muscle satellite cells, which are the dedicated repair crews for muscle tissue, dropping by over 50% by the time these mice are just 5 months old.
5 months? That's barely out of the mouse equivalent of college. But honestly, it grounds the science perfectly. It entirely explains why you can sleep in a slightly weird position in your 30s and wake up with a pulled hamstring that takes 6 weeks to heal. Our repair crews are clocking out early.
It's a great example. And it's not just the muscle satellite cells. They saw tenocytes, which are vital for maintaining tendons, plummeting at that same early adult stage.
Wow.
In the kidneys, there's a significant depletion of podocytes, which are the crucial filter cells that keep the organ functioning. In the lungs, aerocytes drop off. In the ovaries, granulosa cells decline, mapping perfectly onto the timeline of reproductive aging.
So if the functional cells, the filters, the breathers, the repair crews are all vacating the premises, who takes their place? Who are the winners in this biological game of musical chairs? Because nature abhors a vacuum. If a tissue is losing its functional cells, something else must be moving in.
And this is where the dark side of aging really reveals itself. The empty space doesn't stay empty. The study shows that as we age, the tissues essentially get overrun by immune cells. You see a massive expansion of very specific, highly inflammatory immune populations invading tissues where they previously weren't dominant.
The paper specifically calls out plasma cells and macrophages, right?
Yes. Macrophages are essentially the immune system's garbage collectors and first responders. But as we age, they expand almost everywhere. For instance, in the liver, a specialized type of macrophage known as a Kupffer cell sees a huge population boom.
But when you have an army of inflammatory immune cells invading an organ, they don't just sit there quietly.
No, they create collateral damage. They release inflammatory signals that stress out the remaining healthy cells.
But I noticed it wasn't strictly just immune cells taking over. There were a few fascinating non-immune cells that expanded, too.
How ghastly.
For instance, they tracked enteroendocrine cells in the stomach, and those populations expand significantly with age. For you listening, if you've ever wondered why your digestion gets a little wonky or sensitive as you get older, this cellular expansion is a major clue. The physical cellular makeup of your stomach lining is actually shifting its proportions.
When you zoom out and look at these two lists side by side, a very clear pattern emerges. You look at the depleted functional cells, the repair crews, the filters, and you contrast that with the expanded cells, the inflammatory macrophages, the plasma cells.
Yeah.
What this atlas proves is that aging, at its core, is a systemic shift away from tissue maintenance and toward chronic systemic inflammation. The body essentially abandons its own upkeep and instead locks itself into a state of constant paranoid immune alert.
Now here's where it gets really interesting. We've talked about how the body ages in terms of these cellular winners and losers, but we haven't talked about who is aging. I want to highlight a statistic from the paper that stopped me in my tracks. Roughly 40% of these aging-associated cellular dynamics are sex-dependent.
40%.
That means men and women are aging fundamentally differently at the cellular level.
It's a staggering number, and it challenges decades of medical assumptions that aging is a universally identical process. When the researchers isolated the data to look at that massive immune cell expansion we just discussed, they found something remarkable. They discovered a highly female-biased immune activation.
What does that actually look like in the tissues? Are women just getting more immune cells everywhere?
It's a much broader and more aggressive expansion across the board for females. To put the numbers to it, they identified 53 distinct female-biased immune subtypes that expand with age, compared to only 31 male-biased subtypes. So the female immune system is becoming significantly more hyperactive as time goes on compared to the male immune system.
I know they gave a specific example using these things called exhausted T cells. They tracked a marker called GISM-MK plus CD8, and these exhausted T cells expanded heavily in the female lungs and the pancreas as they aged, much more so than in the males.
What's fascinating here is how beautifully this single-cell data maps onto a major epidemiological reality we see in human populations. We have known for a long time that women have a significantly higher prevalence of autoimmune diseases conditions where the immune system mistakenly attacks the body's own tissues.
Right.
What this study provides is the molecular foundation for why that happens. When we see this broader, more aggressive expansion of inflammatory and exhausted immune cells across aging female tissues, we are looking at the exact cellular environment that allows autoimmune conditions to thrive later in life.
It makes complete sense when you look at it through that lens. But this battle of the sexes isn't just about one sex having a little bit more of a certain cell type than the other. I was reading the section on kidney function, and it looked like the male and female cells were doing the exact opposite things in some cases.
Yes.
Can you explain what was happening there?
The kidney data is where you see the ultimate proof of these sex differences. They tracked a very specific, highly specialized type of kidney cell called the proximal tubule S3T2 cell. What they found was that in aging females, the population of this specific cell steadily increases.
Okay.
But in aging males, the population of that exact same cell steadily decreases. They are on completely opposing trajectories.
And it wasn't just the cell counts, right? At the DNA layer, there were tens of thousands of chromatin peaks, the open and close pages of our instruction manual, that were altered exclusively in one sex.
Exactly. And the implications for this are massive. If a cell population is collapsing in men but proliferating in women, it means any universal approach to treating age-related decline is going to hit a brick wall. A one-size-fits-all anti-aging drug might perfectly aid the male kidney while actively harming the female kidney, or vice versa.
So we've got these incredible cell populations shifting, immune cells invading, and male and female bodies handling it all differently. But to me, the biggest question is why? What is actually driving those shifts deep down at the DNA layer? If we bridge back to our instruction manual analogy, which specific pages are being glued shut and which pages are being ripped open to cause all this chaos?
To figure that out, the researchers mapped exactly which regions of the genome were changing their physical accessibility, and the pattern they uncovered is quite profound. If you follow the manual analogy, the pages that hold the instructions for the SOX family of transcription factors, these are the genes that act as the caretakers of the cell, keeping it young, functional, and stem cell-like. Those are the pages that are getting permanently glued shut.
The paper points to specific ones like SOX4 and SOX11. So basically, the instructions for staying youthful and maintaining tissue are physically condensing. The cell's reading machinery literally cannot access them anymore.
Exactly. Meanwhile, the pages containing instructions for inflammation and stress responses are being forced wide open. Regions controlled by major inflammatory regulators, things like NF-kappa B, IRFs, and AP1, become highly accessible.
I saw they highlighted genes like NR3C2 and the inflammatory cytokine IL-7. These become highly accessible across dozens of different cell types. So to put it simply, the cell forgets how to read the manual on how to repair itself, but the chapters on how to trigger a massive inflammatory response are essentially highlighted, dog-eared, and bookmarked.
That's a great summary. But the epigenetic remodeling, this shifting of open and closed DNA, isn't just limited to our standard functional genes. This is where the study uncovered something truly unexpected about our genomic dark matter.
Yes, the waking of the junk DNA. To me, this was the spookiest, most memorable detail from the entire deep dive. As they scanned these millions of cells, they found that highly conserved ancient parts of our genome actually start gaining accessibility as we age. Can you unpack what we are actually looking at here?
It sounds like science fiction, but it's a very real biological phenomenon known as de-repression. Embedded within our DNA are these elements called retrotransposons. You can think of them as ancient viral DNA.
Wow.
Over millions of years of evolution, viruses have inserted their genetic code into our ancestors' genomes.
The paper specifically name-checks subfamilies like line and LTR elements like MLT1F1. For you listening, what this means is that we are all walking around with ancient viral fragments trapped in our DNA.
Right, and normally our cells expend a massive amount of energy keeping those ancient viral elements silenced. They keep that specific section of the chromatin locked down tight, bound up in literal molecular glue. because if those copy and paste viral genes become active, they can cause genomic instability and trigger massive immune alarms.
But as we age, that glue degrades. The silencing breaks down, the ancient pages of the manual fall open, and these retrotransposons become accessible. The cell sees them, panics, and it drives that systemic inflammatory response we talked about earlier.
It is a major hallmark of the aging epigenome, the loss of control over our own genomic dark matter.
So if we take a step back and look at the whole board, we have specialized cells dying off, immune cells invading, youthful genes shutting down, inflammatory genes opening up, and ancient viruses stirring in our DNA. It sounds utterly chaotic. Reading this, I kept wondering, are these changes just random errors accumulating in isolated organs? Like, does the kidney just randomly fall apart at the same time the lung randomly falls apart? Or is there a conductor leading this whole dark symphony?
If we connect this to the bigger picture, the data strongly suggests it is a highly coordinated, body-wide response. These cellular changes are too synchronized across the 21 different tissues to be random local decay. There had to be a systemic trigger.
And to figure out what that systemic signal was, the researchers did something incredibly clever. They took this massive aging dataset they just built, and they cross-referenced it with something called an immune dictionary. How exactly does an immune dictionary work?
Think of it like a forensic database. The immune dictionary is an existing database that catalogs exactly how cells react, how their epigenome changes when they are exposed to very specific inflammatory alarm bells, known as cytokines. Cytokines are the chemical messengers the immune system uses to communicate across the whole body.
So the researchers took the epigenetic fingerprint they found in the aged mouse cells, particularly the B cells, which showed massive changes, and essentially ran it through this forensic database, asking, what does this pattern look like?
Exactly. And the database spit back a perfect match. The aging patterns in the B cells exactly matched the epigenetic fingerprints caused by specific cytokines, predominantly IL-15 and IL-21.
And this isn't just an abstract mouse thing. The paper notes that IL-21 protein levels are actually proven to rise in human blood as we age too. We have the exact same inflammatory alarm bells ringing in our own veins.
This was a crucial validation for the team. It proved that the epigenetic changes they were observing weren't just spontaneous random errors in the cells. They were direct, programmed responses to these specific inflammatory messengers circulating in the bloodstream.
So why is it so important for you, the listener, to care about? Because it fundamentally reframes everything we thought we knew about getting older. It means aging isn't just your local organs wearing out independently. It means aging is a systemic alarm system that gets stuck in the on-in position. These cytokines, like IL-15 and IL-21, are constantly flooding the system, commanding cells across the entire body to open up those inflammatory DNA pages and shut down repair pages.
It creates a devastating feedback loop. As we get older, immune cells expand and release these cytokines. The cytokines alter the chromatin in healthy cells. Those healthy cells lose their function and trigger more inflammation, which calls in more immune cells to release more cytokines. It is a highly coordinated, organism-wide decline, all driven by this systemic signaling.
So what does this all mean? Let's bring everything we've talked about together. What the Rockefeller University team has given us with this 11 million cell atlas is undeniable proof that mammalian aging is a highly coordinated, sex-specific rewiring of our cells and our DNA. We lose our functional tissue cells, we get overrun by inflammatory immune cells, and our DNA physically changes its shape to unleash ancient, repressed viral regions while permanently gluing shut the chapters on stem cell maintenance.
And crucially, we now have the data proving that this entire process looks incredibly different depending on whether you're male or female, with the female body showing a much higher propensity for broad immune activation.
But this isn't just a depressing laundry list of how our bodies fall apart. By systematically identifying exactly which chromatin peaks and cell states are vulnerable, scientists finally have a highly specific map.
This raises an important question about the future of medicine. Historically, treating age-related decline has been an exercise in playing a game of medical whack-a-mole. We treat the kidney disease, then we try to manage the heart condition, then we suppress the autoimmune issue. We're always chasing the symptoms.
Right.
But this atlas gives researchers the root causes. It shows us the exact genetic switches that are flipping at the exact timeline they happen. It offers a genuine therapeutic map to target the underlying epigenetic remodeling that drives all of those separate diseases in the first place.
And That leads to a really profound thought I want to leave you with today. We've seen that aging is essentially our DNA instruction manual opening to the wrong pages, waking up ancient viruses, and forgetting how to read the useful chapters. And we've seen that those pages are fundamentally different for men and women. If that's the true nature of aging, it means the ultimate anti-aging pill of the future probably won't be some universal fountain of youth that everyone takes. Could the future of medicine mean a personalized, sex-specific epigenetic editor? A targeted therapy that doesn't just treat your symptoms but goes into your specific cells and simply refolds your DNA back into its youthful, perfectly tuned shape. It is an absolutely fascinating future to imagine. Thanks for taking this deep dive with us.